Hi, we are Group 20. Our group members are Emir Ergin, Almin Engür, Ata Öztürk, and our project is effects of T2 decay during readout in MRI. What is MRI? MRI is a non-invasive technique that creates detailed anatomical images by detecting changes in alignment of protons in tissues. Strong magnetic field align hydrogen protons in the body. A radio frequency RF pulse excites the protons and magnetic field gradients encode spatial information. The resulting signal is detected by receiver coils and stored in K-space. Using Fourier transform techniques, this data convert into the detailed images. What is time intervals T1 and T2? As we know, TR is a repetition time is the time interval between consecutive RF pulses. The interval between the RF pulse and the signals collection is known as the echo time, which is TE. After RF pulse is turned off, the protons return equilibrium point and leading the type of relaxation. One of them is longitudinal recovery, T1 relaxation, recovery of the magnetization along B0 axis, and T2 relaxation, which is transverse decay, loss of coherence in the transverse plane due to spin-spin interaction. As you can see from the figure 2, the different tissues, white matter, gray matter, CSF, has different type of T1 and T2 relaxation. What our project is about that we will investigate the T2 effects, T2 decay effects during readout in MRI. What we're gonna do, we will change the readout window size to observe the T2 decay, changing the resolution and observing differences between ideal and non-ideal case.
So as our data source, we use the brainwave database, which offers high-resolution 3D brain models. From this database, we were able to assign unique T2 values for each tissue, and we were also able to detect the slice we want to use, but for the simplicity, we kept the slice constant. So now I will talk about the tissue-specific T2 decay modeling. The tissue-specific values were assigned to the brain images downloaded from the brainwave, and they were ranged from 0 to 10. Based on these data, we were able to assign unique T2 values to each tissue, and we constructed the T2 map. Also, since we were able to obtain the T2 values and proton density values from brainwave, we were able to calculate the M0 values. As you can see from the table, the T2 values and proton density values are available. T2 weighted signal was computed for each voxel in the brain model at echo time. The calculation is done according to the formulas you can see. The formulas were obtained from lecture notes, which represents the T2 effects until the TE time. For the case-based simulation with T decay, to simulate MRI acquisition, the weighted T2 weighted brain image was transformed into case-based using the 2D Fourier transform. So for each value of T, corresponding column of M is acquired from the Fourier transform of the multiplication of exponential term and the signal calculated before at TE. So T here represents the elapsed time during the case-based acquisition. And M here is the case-based representation of the transverse magnetization, and exponential term implements the effect of T2 during the case-based acquisition. So for the image reconstruction, image is reconstructed with inverse Fourier transform. For our resolution, the original brain image was sampled at a resolution of 1 to 1 millimeter. And to simulate different resolutions, we resampled the images by using the bilinear interpolation. For the simulation of multiple readout durations, for each readout value, a time vector was defined, and T2 decay is applied in the time domain for each time space. The Fourier transform of the decay images are taken. The columns corresponding to the time steps were taken in K-space from the transforms, and finally the image was reconstructed by taking the inverse Fourier transform of the K-space acquisition. For our simulation of multiple resolutions, first resolution is defined for each resolution. Image is resampled by using bilinear interpolation, and the same steps are done which was mentioned before, like case-based simulation, image reconstruction. So I'm now going to talk about our assumptions and limitations. So our readout window size is extended to observe the pronounced T2 decays, and noise was added. And we also changed our resolution to study the impact on 2D decay and image. For the ideal case, we did not use any noise, and readout window is kept very short. During the readout, we did not implement the 2T decay effect. And for the resolution, we used a fixed resolution, which is 1 millimeter to 1 millimeter. For the both cases, we used TE as 100 milliseconds.
I will demonstrate the implementation of the simulation on MATLAB. Here, as you can see, this is dimension for the dataset. Here are the resolutions we use. We implemented for 5 readout durations, which are 10 milliseconds, 45, 80, 250, and 500. And since we implemented for various results, I will talk about the results on the presentation part. Here are the significant results we chose to demonstrate. The first table represents 10 milliseconds and 500 milliseconds as an extreme case. And also, this is the case where we take T2 decay at readout duration is ignored. And here are the same results for with the absence of cerebrospinal fluid and skin's proton density value. We also did this because of the high T2 value of these tissues. The effects are hard to see in this table, so we implemented as this as well. And as you can see, with lower resolutions, the effect is more clear, and here you can see artifacts occurring on the image, and for 3 millimeter resolution, the effects are significant as ringing artifacts occur as well. As discussion, differently from the lectures, we looked at a non-ideal case possible to occur in real life, which is the effect of T2 decay during readout phase, and we didn't discover it in detail in the lectures. Also, we differed resolutions to compare different scenarios. Also, we added noise to the image to make it more realistic. We saw that as readout window increases, the effect of T2 decay is more significant, and also lower resolutions causes higher T2 effects. To improve the results, we could further add the field inhomogeneity and motion artifacts to make it even more realistic. Here are our references, and thank you for listening.
Thank you.